Open the pod bay doors, Hal.
I'm sorry, Dave. I'm afraid I can't do that.
What would you do with a brain if you had one?
Do? Why, if I had a brain, I could...
Miles per hour.
I could while away
46, 56 degrees.
Thanks, everybody. It's really great to be here. Good afternoon. It was a beautiful day. So we appreciate you taking the time to come in and hear us talk today. So we're going to talk about forensic science and a bunch of issues. And as you can tell from the title, there are some issues with forensic science, and we'll get into the details of what that exactly means, and talk about a lot of the science we're doing at the lab to try to improve that and make it better. So specifically, we're going to talk about what is forensics. What do we mean when we say that? Because different people mean different things, so I'll tell you what we think it is and how we're approaching some of the issues. We'll talk about evidence types, and how is that currently done in a law enforcement context, law enforcement community, and police, how do they use forensics in court. And then if we have those methods already, then why do we need research, and what are the problems that we're doing? And finally, what are we doing at the lab to focus on issues related to forensics that I'll talk about? Again, if you can tell from the title of the talk, it has to do with protein. So along the way, we're going to talk about what is DNA, what are proteins, and how do all these things weigh in. So the movie you're going to see today is Gattaca. Has anyone seen Gattaca? Okay, none of you are allowed to spoil. So it's a great movie, and so the first thing I'll point out is the title. Okay, so as we walk through this talk about some of the science. We'll talk about the title of the movie and how that's important, what that represents with respect to forensics. Also, want you to think about this movie in the sense of when it was made. The movie was made in 1997, so 20 years ago, which it seems like yesterday to me, unfortunately. I guess that was a big one. So 20 years, so you'll be able to see where we are now compared to what at the time when they made the movie was thought of as being very futuristic, sort of things that were so far off in the future no one had even thought of. This was all really new stuff. So pay attention as we're talking through the science, and when you watch the movie, hopefully it'll trigger some things back to what we said during the talk. So let's get into this a little bit, talk about what is forensics. Okay, so if you look up on Wikipedia, which is what I always do when I need to understand what something is, then what you'll see is forensics is the application of science to criminal or civil laws, okay, and civil laws, I guess. And so we think about this in the context of like TV shows like CSI, right? Crime Scene Investigators. So it's a process of going to a crime scene, you collect evidence that could be associated with the crime, you typically take that back to a laboratory and you make measurements. You try to understand, what does that forensics mean? Can I associate an object with a person or an event, like a crime? And then that information is used in a court of law to determine primarily is someone guilty or innocent of some activity. Okay? And there's a lot of different types of evidence. The process itself is pretty straightforward. It's evidence collection. This is what you see in the shows where they go and they take fingerprints off of door jambs or car door handles, or they collect the bloody knife, or they collect a swab of blood. That's the evidence collection part. It's very important. There's evidence preservation. This is really key because in order for accurate scientific measurements to be made, the evidence has to be stored and protected in a way that it's not going to get contaminated, for example, or degrade in a way that won't allow it to be measured. So that's really important, that evidence preservation. We're not going to talk about any of that today, really. What we're going to talk about primarily is the analysis and how do you actually analyze evidence and what methods are used. And the key here is this is where it really differs, reality differs from what you see in TV. I think these measurements don't happen in 20 minutes with commercial breaks. It doesn't work that way. And oftentimes, you'll see in these shows, they say, oh, we found this and that proves definitively that this was the person. And put them away and they go to prison for the rest of their life. The problem that we have is that a lot of the evidence we use doesn't allow that kind of precision. So that's some of what I'm going to talk about today. The final step in forensic science is the presentation of those results. And so most commonly that's done through a report, or what we mostly think of is someone sitting on a stand in court and testifying to the measurements that they made. So let's talk about some specific types of evidence that we're familiar with. So in general, this is, we're talking about pattern evidence, okay? So fingerprints, that's a great example of pattern evidence, okay? So fingerprints, or finger marks as it's often called in the law enforcement community, are left at crime scenes. We all have sort of what we assume to be unique fingerprints, although, as you'll hear me talk about, that's not something that's has been proven, or is there really a way to prove that, unless you go and look at everyone's fingerprints. And what they do in fingerprint examination is they go around and they look at a fingerprint and they look at all these features. You can see there's things they call crossovers, where you have ties between ridges. There's the core and sort of what that looks like. You have places where ridges split apart. Those are bifurcations. You have highlands and all kinds of features on these fingerprints. And so an expert, someone who's been trained, has been determined to be an expert in fingerprint examination, will look at the fingerprint and they'll do what they call encoding. They'll identify all these features and they'll make a list, essentially. And they'll use that list to compare to other fingerprints, either to known people or to other fingerprints left at a crime scene. But it's very, very complicated to do, and it really relies on the expertise of the individual doing it. No two examiners are going to give you the same exact result of doing this sort of analysis, and that's a problem. What that means is that type of evidence is subjective. Okay, it's based on a human's interpretation of what they're seeing. Okay, so what other kinds of evidence do we have? We have firearms and bullets. Okay, so in these cases, often what they're doing is they're measuring bullets, they're making sure the calibers are right, and then they're looking at things like how the rifling of the bullet leaves marks on, rifling of the gun barrel leaves marks on the bullet itself. They're looking at firing pin marks left on the casing and other marks left by the machinery of the gun on casings, and they're trying to match that back. It's another example of subjective evidence where you have an individual who's been trained and he's making comparisons between objects that he's looking at. Okay, these aren't scientific measurements, but they're observations, observation-based analysis. And now a really important one we're going to talk about is hair and fibers. And really, by hair and fibers, we mean hair. Okay, so this is when hair is left at a crime scene. And I'll give you a hint, this is really important in the movie. We all leave hair, it's all falling off us all the time. We shed skin cells, all this stuff is being left in the environment as we sort of passively go throughout our day. So what a crime scene examiner does, or a hair examiner, is they'll actually put the hair under a microscope and they'll compare it to another hair directly. And they'll do things like measure the width of the hair, the pattern of the external markings and scales, and various other features of that hair, and make a determination about whether two hairs match or not. Well, that's going to be really problematic because we all do things to our hair. Some of us do things to our hair. We dye it, we curl it, all those sorts of things. Sometimes hairs can be very similar. It can be very difficult to accurately do a hair comparison. And again, this is very subjective. It's based on a person's interpretation. Okay? So all of these types of evidence I just talked about are all called pattern, like I mentioned. And there are a bunch of other types too. Right? There's tool marks. If someone jimmies your door open, they're going to leave specific marks from the tool. There's tire tread analysis, shoe prints, bite marks, all kinds of things where people are leaving marks or indications on evidence related to a crime. All of these are examined by an expert who's been trained and coming up with an opinion, and making a decision about whether there's a match or not a match between two pieces of evidence. But there's another type of forensic evidence also, and this is called analytical evidence. And so these are methods that are based on scientific measurements. In these cases, the person who's being brought out of that loop, they're not making an opinion or making a decision, they're making a there's a determination made about the evidence based on technical measurements that are made using instruments such as the one shown here. So a good example is chemical analysis. So if we find in someone's trunk a bundle and we open it up and it looks like cocaine, well, is that sugar? Is it cocaine? Is it something else? How do we know that? Well, we use analytical instrumentation such as this. This is a liquid chromatograph, mass spectrometer. And we can put that sample in that instrument, and that instrument can tell us, based on comparison to standard known cocaine, for example, that that is cocaine and not something else. That's not someone's opinion. That's an objective piece of information. There's no human bias inserted into that decision. Okay, so that's really important. And so there are other kinds of objective or analytical forensic evidence as well. So blood type, this is one we're pretty familiar with. Everybody's got a blood type. We inherit it from our parents. It can be A, B, O. It can be positive and negative. And so what this can do is it can tell us that blood from a crime scene, for example, is consistent with the blood type of a perpetrator. But that's a pretty small number of options, right? There's only a handful of options that we all can be. And so there's no way to say definitively that the person that left that blood, even if the blood type matches, was from them. So although it's objective and it's definitive as to that match, it's not exclusive to that individual. So there's no way that we can pin that specifically on that individual. Okay, so now we get to something that is more specific, and that's DNA. This is something I'm sure you've all heard about in the news or on the different shows. So I want to talk for a minute about DNA. So what is DNA? DNA sits, and nuclear DNA in particular, sits in the nucleus of all of our cells, and it's contained in chromosomes. And we inherit our chromosomes from our parents, half from one and half from the other. And they're mixed together, and that mixture provides us with basically a recipe for who we are, the blueprint for all the proteins and machinery that's built into our bodies, that's unique. It's like your mother told you, everyone is special, everyone is unique, and it's because of DNA, okay? And so DNA, the molecule, is actually pretty simple. It's a very long molecule, and it's made up of 4 simple building blocks, and they're shown here. C, G, A, and T, okay? So now I'm going to give you a clue. If you look at those 4 letters, and we abbreviate those based on the first letter of the name, so C, G, A, and T, cytosine, guanine, adenine, and thymine. Look at those letters and then think about the title of the movie, and it might give you a clue as to how they came up with the title there, Gattaca. Okay, and so the sequence that these are incorporated into our DNA, that pattern, is what tells our body how to make proteins. It gives us the instructions. We have machinery within our cells, every one of our cells, that's able to turn that code into a protein product. And that's ultimately what we are composed of. And so when Dr. Mason comes up here, she's going to talk specifically about how that process happens. It's really fascinating. But essentially, it's come together to form things called genes, and these genes are what are responsible for forming individual parts of our body that have functions, okay? And so how is this used in forensics then? So let's think about this. So let's just say we've got Mr. Sunglass Guy here, and this is his DNA. Okay, the yellow boxes represent the genes in his body that tell the machinery to make something. They say, here's how you make hemoglobin. Here's what color your hair should be. Those sorts of things, okay? And the reality is, even though we're all very unique, the genes that we all share vary very small amounts between each other. So there's only about 1% difference between all of us in our genes. Most of our DNA is the same. So because of that, they're not very useful at individualizing people. They're not very useful in forensics when you want to identify a specific person. But lucky for us, as forensic scientists, there's a ton of DNA that sits in between these genes that we used to call junk DNA. We thought had no function whatsoever. It was just there as filler. The reality is now we know that there are some functions associated with that DNA, and it does give our body instructions about when to turn on certain genes and when to turn them off. But importantly, they also contain something that we call short tandem repeats. Okay, STRs for short. And what these are is they are regions within this DNA where you have short sections. So in the case of the blue STR, the short DNA sequence is GATA. Okay, and the green one shown over here is AGAA. Okay, so those are regions where that same short 4-base sequence repeats over and over again. Okay, and so everybody on the planet has these regions, and they have some number of these same sequences, but the number of times they repeat varies hugely among the human population. And so what we're able to do is we go in with our instruments and we count how many times does that sequence repeat in person A versus person B. Okay, so what does that look like? Let's say we had 2 people. We had one crime that was committed. We measured the sample from the crime. And we've got 3 suspects. Okay, so in this case, Mr. Sunglass Guy has these 2 STRs. One repeats 12 times and one repeats 3 times. Mr. Mustache has the first one repeats 4 times and the second one 15 times. And then Carrot Top here, the first one repeats 9 times and the second one 8 times. Okay, so you can see that they're different. And our ability to measure those allows us to compare them to a sample from a crime scene. So let's say that the crime scene had DNA that had 9 repeats in the first section and 8 in the second one. That would mean it would be a match for Carrot Top, and these guys can go free, go have lunch or something, okay, and let them go. And And so that's fundamentally how this is done. The real power, though, here is that there are a number of these. There's between 13 and 20 of these regions that we can measure. And by using those together, we can get ridiculous power. It's called power discrimination. So the number that we can achieve with DNA is called 1 in a quintillion. That's a 1 with 18 zeros after it. It's a huge number. It's almost meaningless. It's so big. So there's 10 to the 10th people on the planet about. So this number is, the reality is that's the population of the planet for every person on the planet. It's a very big number, right? So the long story short there is that with almost perfect exclusion, we can eliminate other people if we have a DNA sample to compare to, and it's in good shape. Okay, so that's why DNA has been so powerful. Now, I mentioned at the beginning of the talk that forensic science is in a state of crisis. And that's because, although DNA is awesome, we don't have it very often, and we're very reliant on subjective methodologies, the pattern evidence I talked about before. And that's very problematic. And so DNA has had a huge impact on this. A couple of examples. Has everyone here heard of the Innocence Project? It's been in the news quite a bit. Okay. The Innocence Project is a group of primarily lawyers and scientists, and they go around and they find people that have been convicted of a crime and using subjective evidence, but they suspect they may be innocent, and they use now available DNA techniques to either prove their innocence or their guilt. And what they found, and this data is now a couple of years old, but so as of a couple of years ago, their results using DNA had resulted in the exoneration of over 300 people that had been wrongfully convicted of a crime. Combined, those people had spent over 4,000 years in prison. It's a big number for people who don't belong there. And the other piece of that is if they had found the actual perpetrator in those cases, then they could have prevented about 130 violent crimes by putting the correct person in jail. Okay, so evidence is really important, right? The scientific evidence and having confidence in that evidence is important because we want to put the right people in jail and we want to keep the innocent people out of jail. Fundamentally, everybody agrees on that. I got another example of how subjective evidence has been problematic. So this was a study done by the FBI about a year and a half ago, where they went back to 268 cases where there had been convictions based largely on hair evidence, the subjective kind, okay, where two hairs were compared under a microscope, an examiner determined that they were a match, and that person went to jail, okay? So in 268 of those convictions, they went back and looked at DNA evidence now available from hair, and they found that in 257 of those cases, that hair evidence was incorrect. That's over 95% of those cases that were based on hair evidence were wrong. That's a big deal, right? Think about that for a second. 257 people were put in jail in part based on hair evidence, and that hair evidence was incorrect. What's even more troubling about that is, so only 11 of those cases were correct. In 32 of those cases, they were death penalty cases. Okay, so the consequences are enormous. About 18 of those 32 cases, the person actually died in prison. Either they were executed or they died in prison because sometimes that happened in prison. So the consequences are huge. And the bottom line is we can't rely on subjective methodology anymore to convict people when the stakes are so high. Okay, and so what you found now is that hair evidence isn't used very much in court anymore because of these results and other things. So we're sort of losing forensic techniques as we go along. And that's really why we're in a state of crisis. We need better forensic methodology. So we have DNA, we're done, right? We've got our solutions. The problem is DNA isn't. It's great, when you have it, it's the best thing to use, but the reality is DNA is very hard to get from a crime scene. And this is the other thing they don't tell you on the TV shows, is that the success rate of getting a full DNA profile from a crime scene or an object associated with a crime is in the low sort of single digits, okay, mid to low single digits. So, in very few cases do you actually get usable DNA from crime scenes. When you get it, you're good, but in most cases you can't have it. And that's because of a number of reasons that we won't go into a lot of detail here. But this gets us into what are we doing about this, okay? And so the reason I brought up so much about the hair cases is that we decided to focus on hair evidence as an area where we might be able to work, okay? And so what we're going to talk about today is a new approach to human identification. And it has the power of DNA the sense that it has very high probabilities for matching. It's a science-based, objective, non-biased method to look at the evidence, but it doesn't require DNA to be present. Instead, we're using proteins. And so I'm going to invite up my colleague, Dr. Caitlin Mason, and she's going to tell you a little bit about how DNA turns into protein and how we're using that protein information to get great forensic results.
So how can we use proteins to identify humans? Well, the answer is here in this example. As Brad explained earlier, your DNA is what makes you unique. One of the reasons why this is because the specific code that your protein has, or your DNA has, tells your cells which proteins to make. How this works is that each set of 3 nucleic acids, called codons, tell your cells in a specific pattern which building block to combine together to create a protein. In this example, you see that a GTT codon next to an ACC codon tells your cells to link the amino acids valine and threonine to one another. Once your amino acid chain has been created, you now have a protein, and for example, hemoglobin. The bonds that link amino acids to one another are very stable. They have the tensile strength of a strand of silk and are almost an order of magnitude more stable than the bonds in DNA. Because of this, proteins can persist at crime scenes for longer periods of time and are easier to collect as evidence. As Brad mentioned before, we're focusing on hair. Hair is an excellent candidate to apply our new technology because it is a type of evidence that is often found at crime scenes. It is composed to up to 90% of protein. And he mentioned hair and fiber analysis is very flawed, so creating a new relevant technology can help add to the forensic science toolkit to gain relevant forensic information from hair evidence. So how can we use proteins to actually identify a human? Here's the example I gave you before. The GTT codon next to the ACC tells your proteins, or your cells to create a protein with valine and threonine next to one another. But what happens if a particular human has a mutation in their genetic code? As you can see here highlighted, the C nucleic acid has been mutated to an A nucleic acid, changing the codon pattern. In many cases, this mutation in your DNA tells your cells to add a different amino acid in the protein chain. So here you can see the threonine is now encoded to an adenine residue. So combining these mutations found in proteins can give us clues as to what person the protein evidence belongs to. So now that we understand that mutations in DNA can result in mutations in hair, how do we use these mutations, specifically in hair evidence, at the molecular level? First, we must consider the molecular structure of hair. Keratins are the class of proteins that largely make up hair samples. And they orient themselves in long strands that bind to one another by isopeptide and disulfide bonds. These strands are wrapped around one another and create a complex structure that yields a hair filament. So before we can look at specific mutations in individual proteins of hair, you have to break the bonds that make hair so stable. The technique that we use to do this is called proteomics. The first step is to take a hair and break it into its individual protein components. But proteins are too big to analyze specifically, so we do a further breakdown of hair by an enzymatic reaction that essentially cuts our hair proteins into small segments called peptides. The resulting peptide mixture is too complex to analyze just as itself, so we separate them in solution using liquid chromatography. As the complex mixture of peptides that are separated are eluted into a mass spectrometer, we can use a combination of bioinformatics and protein databases to determine the specific sequence of the peptides and therefore the specific composition of each protein. To explain this in detail, Dean will now take over.
Okay, so Brad and Caitlin showed you how protein evidence can be used for individual identification. And this is done by finding what we call identity markers, which are mutations in DNA that are reflected by amino acid changes in the protein. So what I'd like to do right now is spend a little bit more time discussing how, in the laboratory, we find out if a particular individual has one amino acid or another amino acid, and how we use that change in amino acid as an identity marker. So I'm going to start with just one of the keratin proteins that are found in hair. This is keratin 39. It's a protein that has a change of 491 amino acids. And so, as depicted here, each amino acid is given by the single letter abbreviation. So you can see, if you look up there, you'll see 491 letters. Each one representing the chain of amino acids that are in this protein. And this protein is just one long chain, so where it changes from line to line, those are all connected together. And this protein is wrapped in the fashion that Caitlin described to make up a hair filament. So when we do the extraction, we get proteins out. As Caitlin mentioned, our instrumentation can't really look at an intact protein to find out the amino acid sequence. So we need to break it down into peptides. And the way we break that down is using enzymes to do a digestion, and the enzyme we use is trypsin. And so if you look on the top row, you can scan down there, you can see letters K that represent lysine, and letters R that represent arginine. And trypsin breaks up the protein after every K and R that's in there, every lysine and every arginine that's in there. So the next slide will show you where these breaks occur according to the peptide. So now, instead of a single protein, it's now broken down into 45 peptides that have 3 or more amino acids present in them. And so we've done two things. We've broken this protein down into peptides, which our instruments can now look at, and I'll describe to you in just a few minutes. It can determine the sequence of amino acids within those peptides and determine if there's an identity marker present or not. The other thing it's done is it's made our job much, much harder. So I'm showing you one protein here. The hairs basically have hundreds and hundreds of proteins in them, upwards of 300 or 400 proteins present. So you can imagine if you take those 300 or 400 proteins, you digest all of them up into roughly 50 peptides, you now have a huge mixture of peptides that you need to analyze. And so what I'll do is I'll describe a little bit how we do that analysis. And part of that analysis, we'll use a particular peptide that I've indicated here. And so we're going to focus in on this on future examples. And basically, I'm going to describe to you how, for this particular peptide, we're going to be able to figure out the sequence of amino acids that are present there and determine if there's an identity marker present in that peptide. And so basically, the instrument we use is It's called the Nano liquid chromatography method attached to a mass spectrometer. And in a nutshell, what this does is the chromatography system separates these peptides out so that we can now look at the peptides individually at the mass spectrometer. And the thing you can think about what the mass spectrometer does is it essentially weighs each of those peptides. It not only weighs it, but also breaks it apart in a particular way. We can weigh the fragments. And again, I'll talk to you a little bit more in detail about how that's done. But first, let's look at the individual components here and understand how they come together to allow us to do a peptide analysis. So the first part is the nanoliquid chromatography system. So many of you in your maybe elementary or middle school or even high school classes may have done paper chromatography. And in these experiments, you may have taken a strip of paper, put a drop of ink at one end, and then dipped the paper in such a way so that water moved up the paper and ink separated out into different colored components. We're basically using that kind of approach to separate our peptide mixture up into all of its components. And so instead of paper, what we have, and it's hard to see on this slide, but there's a small column here which kind of takes the place of the paper. And the idea is that we put our sample at one end of that column. Instead of letting liquid wick through, we actually pump it through with some sophisticated pumps that are here. We push the peptide mixture through the column, and the different peptides travel through at different rates. Okay? And so here at the end of the column, the peptides are coming out a few at a time. And they're coming out at a few at a time, and our mass spectrometer can now analyze them one at a time as they come out. The part of the system between the liquid phase here, where our samples are being separated, and the gas phase here, which is where our analysis is done, is called a nanospray interface. And so here, what we need to do is take the peptides that were in a liquid environment and do two things to them. First, we have to get them into the gas phase, because our mass spectrometer will use analysis in the gas phase to do its measurements. And the second thing is, we have to put a charge on each of those molecules. So now it's a charged molecule, which is known as an ion. And so these ions can be manipulated by electric fields. And so the mass spectrometer itself is using the charges and the masses and electric fields to determine what the mass of that ion is. And so this particular mass spectrometer we use is a hybrid quadrupole Orbitrap mass spectrometer. And so this instrument does two things. It can measure the mass of an individual peptide that comes in. And it can do that at very, very, very high resolution. And so based on the mass alone, we can almost guess exactly what the peptide content is. We don't know the sequence yet, but we can make a pretty good guess about what the amino acid content is. The second thing it does is it takes those individual peptides and it breaks them up into fragments. And I'll give you some details on how that breakage is done. But basically what it does is it takes the ions, puts them in an electric field, so it accelerates those ions into a gas cell that then breaks those ions into its fragments. And then it also measures the fragments. So you can kind of see our process for doing protein identification involves a lot of breaking of things, right? We break the proteins down into peptides, and we break the peptides down into its fragments. And then once we get to that level, we can tell what the amino acid sequence is in each peptide. We can then build back up what the protein must have been and basically work backwards from there. So this is an example of what the data look like coming out of the mass spectrometer for a single peptide. And there's a lot going on here, but what you're seeing is along this axis is the mass to charge ratio of the ion. So you can think of this as basically the weight scale, if you will, of the ions themselves. And this vertical axis is basically just the intensity of each ion. Now there's a lot going on here, but what I like to do is basically simplify this by looking at one series of peaks that are here. So you can see there's some orange peaks that have Y labels on them. So here's Y10, Y9, 8, 7, 6, on down. So what I'm going to do now is show you a new slide where I've taken everything out except for those orange peaks. Okay? And so what we're going to do is look at this particular series and understand what this is telling us about the peptide. And so basically what we've done in the mass spectrometer, we selected this peptide that I pointed out earlier. We've taken it and we've accelerated it through the gas and broken it down into fragments. And what happens when you break it down into fragments, which is pretty lucky for us, is it tends to break between amino acids. Okay? So if we take this particular ion here and we collide it, it may break at this bond. And what you get when that breaks is a ion that is now differs from this one by just one amino acid. So you may be able to see where this is going, right? That the difference in weight between these two is the weight of this amino acid. So we can do an individual amino acid identification that way. You know, kind of a simple analogy to this would be, you know, if you had, say, a big truck with 4 heavy crates on it, you want to know the weight of each of those crates, right? One thing you can do is take each crate off and weigh it one at a time, or you can drive the truck onto a truck scale, and you can take the crates off one at a time, and you would see the weight difference every time you took the crate off. And you can deduce how much each crate weighed. And so that's kind of what we're doing here. And so we're getting you know, fragments that differ by one amino acid. And we label these amino acids with a Y because it's just convention. It's keeping this end of the peptide, and then the number is the number of amino acids remaining. And so 12 amino acids represents the intact peptide that we fragmented in the first place. Y11 is if we break this first bond. And you can probably guess where this is going, right? If we break the second bond, we get a peptide that's one shorter, that's Y10. And this is the first one that now shows up in our mass spectrum here. And so here's Y10 in the mass spectrum. It's the highest weight of all of these. Again, we just go down the line. We break the next bond over. That's Y9 that appears here. We break another one. That's Y8. Probably seen a pattern here by now, right? That's Y7, and so forth. And so what we have are a whole series of peptides, and each of those peptides differ by one amino acid. And so if we know the differences between these peaks, we know the mass of the amino acid that just came off. And most amino acids have unique masses, and so if we know the mass, we know the identity of the amino acid. And so, for example, you know, if in the Y8 we didn't know what this one was, you know, what the identity of this amino acid, we would simply take the difference between this mass and this mass. This is kind of like, you know, taking one crate off the truck and weighing it. The difference there is the difference of this amino acid. And so, what we'll find out is that in this peptide, most people perhaps would have a tryptophan in this position, but some of the population might have a cysteine in the same position. And so this is the amino acid substitution that Caitlin was talking about that reflects a mutation in the DNA that changed the codon and so forth. And so one way to think of what we're looking at here is really an echo of your DNA sequence showing up in proteins. And so now we can use peptide identifications relative to whether we have, say, the normal peptide or the variant peptide as a way to identify a person. Now, you may ask, how many of these markers do we need to uniquely identify one person? So one marker isn't enough, right? Because, you know, we may say, for example, just to keep the math easy, 50% of the population had the normal one and 50% had the variant. Well, if you find that variation, well, you can only say, you know, half of the world's population is consistent with your evidence. And that's not very, very discriminating. So what do you need to do? So what we do is we work our evidence and calculate the power of discrimination based on probabilities. And these probabilities are based on the product rule. And so the idea here is going back, you know, let's say we have a population of people here. We just represent that with the beginnings of a pie chart. And let's say that we have one marker, as I said, at 50%. Well, that only discriminates half of the population, right? Well, let's say we had a second marker, and that marker was also available at 50%. So you can ask yourself, well, how many people would have both markers? Well, half of the people would have this marker, also have a marker, and so also have the second marker, and so 50% or 25% of the population would have both markers, right? And again, you can kind of see where we're going now. If we have three markers, Well, half of this quarter would also have that third marker. And so if you have 3 markers, you can discriminate in 1/8 of the population. You can keep going. So every time you get a 50% marker, you get a little bit more discriminating. And so if you multiply this out, if you had 10 markers, you would have 1 in 1,000 discrimination. If you had 20 markers, you have 1 in 1,000,000, and so forth. So this is kind of a simple example where you can kind of do the math in your head. When you look at actual identity markers that are present in proteins, they have different frequencies in the population. They're not all 50%. And so what we've done is some statistics to kind of look at how many markers would you actually need. So this chart here has a number of markers that you might need and the amount of discrimination you will get from that number of markers. And so if you look up here, 7 billion or so is the population of the Earth. If you map over here and down, you need in the ballpark of 100 markers. Okay? And so I've already showed you our instrumentation that can take hundreds of proteins, each broken down into, say, 50 or so peptides. Well, if we just need 100 of those to look at, that's well within the range of our instrumentation. Just a couple of other benchmarks on this graph. The population of the U.S. is a little above 300 million. If we map this over and back down, you need about 80 markers to be unique amongst the population of the U.S. Where we are right now, we have in the ballpark of 60 or so, and so we're kind of at the 1 in a million mark. And so our goal right now is to go through lots of hair proteins, find markers that could be candidates for identity markers, make sure that they're useful in a forensic setting, meaning that they have frequencies that are useful, that they are persistent in the environment, are easy to detect. And so we're going through that process of this discovery to get candidate markers that then build into a panel of markers that can be used in a forensics lab. So far we've really focused on hair proteins, but as you know, proteins are found in a lot of other tissues in human bodies, and so we're going to look at a couple of cases which are of particular interest in forensics. One is teeth, and so you may find a skeleton that has either been in the environment for a long time, or it's been in some sort of harsh environment like a fire or something, and so the DNA has been destroyed, but may still be able to get protein from those kinds of samples. And one area where you might look in a skeleton, of course, are in the bones. And so we can take bones, extract the proteins out of the bones, and do the same approach for finding identity markers in bone proteins. We've also looked at teeth, and what's kind of interesting about teeth is there's a junction here between the enamel and the dentine that has an enzyme that's responsible for mineralization of your teeth. And that's a protein called amylogenin. What's interesting about amylogenin is that it has 2 different types. One that is coded on the X chromosome, and one's on the Y chromosome. So we can look at the types of amylogenin that are present in teeth and determine sex of the skeleton. And so this is really important because if you have a partial skeleton, but you do have a tooth from it, you can determine the sex that's of the skeleton itself. The other interesting thing about the bones and the teeth are that these are often found in archaeological settings. And so if you think about archaeology, that's really just like forensics, right? So in modern forensics, you have a crime scene, you're trying to find evidence to put together a story about what happened there and like people and activities. Well, archaeology is really the same thing using ancient samples. If you find a burial site or a dwelling site, you find evidence there and you try to figure out who was there, what were they doing, and so forth. And so a lot of the applications that we're working on right now or a lot of the developments we're doing right now can be applied to archaeological setting as well. So just to wrap this up, we're looking at protein evidence to associate a person with a crime, either a scene, an object. And what's really important is that we really see these protein techniques folding in with other types of evidence that you find, right? So there may be DNA evidence present, but you can augment that with protein evidence and other types of pattern evidence. For unidentified remains is really important for missing persons. that it may have been in a location and left, say, hair or skin evidence behind. Also mass casualty situations where if there was some sort of disaster where a lot of the forensic evidence has been destroyed, you need all the evidence you can get to make identifications. And as I mentioned too, looking at archaeological samples, because the proteins are related back to DNA, you tell a lot about the background of people and look at migration patterns, when the first contact occurred between groups, and also sex determination, so you can understand some of the cultural differences around sex and gender and things like that. So, with that, I'd like to thank you for your attention, and we'd be happy to answer any questions. Yes, sir. Okay, so the question is about fingerprinting, and has there been any automation in fingerprinting? Can I say something?
To some degree, so what you've seen for automation there is the comparison process. So once the fingerprints have been encoded, it gives you essentially a list. And those can be, so you'll see this accurately portrayed. It shows a lot of times it'll show fingerprints going through a computer to be compared. The actual process of encoding is still a manual process. There's an examiner that does that. And so there are people that are looking at ways to do that and kind of standardize that approach, but we're not there yet.
If someone else has a question, I'll run the mic to you. And please ask. When do you ever get to ask a question of a Lawrence Livermore National Lab scientist? This is a rare opportunity. Oh, right here. I'm coming.
I'm just interested in how long the teeth are dental, and is there a registration among dentists, and how has all of this sort of come together with law enforcement? You know, that whole registration and connection.
Yes, so that's a great question. And we're kind of where DNA was, maybe 30 years ago. We're just working on kind of the science side of it. We're at a stage right now where we can basically exclude individuals, and so we can say this is not a match. Inclusion is much harder, right? And so we're working on getting more identifications. We're also working on publishing our work so it gets accepted in the scientific community. But I think also you might be alluding to, there are also ethical questions related, right? Because as was the case with DNA, as you start collecting biological material, you're not just collecting identity information, you're also collecting information might be related to health and so forth. So lots of kind of interesting, medical ethical questions and privacy questions as well. But in terms of a timeline in using this in a forensic setting, it's kind of a couple of years out yet before you'll see kind of inclusion in courts of law. I think eventually there will be kind of databases of protein markers, and individuals will be, just in the way DNA is right now. When a person is convicted of a crime, their DNA goes into a registry. There'll be similar registries for protein, but we're a ways away from that right now.
I have a question. What is the difference between MALDI-TOF used in the clinical laboratory and the forensic methods that you're using?
Yeah, so MALDI is a technique. For those of you who have never heard of MALDI, it's a technique for mass spectrometry where instead of introducing the sample with chromatography and nanospray, as I described, you use a laser. It's matrix assisted laser desorption ionization. And so this technique, you take samples, you put them on a plate, you use a laser to both ionize the molecule and send it into gas phase. And so this is a common technique used for protein analysis. We're using, you can certainly use MALDI for this kind of work. I think, we're using the chromatography mass spectrometry just because we have such complex samples. In the future, I think where you're going to go in terms of routine use of this technique in a lab will involve liquid chromatography and triple quadrupole mass spectrometers. These are the type of mass spectrometry that you might find in a crime lab, let's just say toxicology testing on samples. And so we need kind of high-end mass spectrometry to do this discovery of markers and the scientific and statistical work that goes behind getting an identity panel. But when it goes out into the fields, much simpler kinds of techniques will be used.
Someone else have a question? I'll run the microphone. Okay. Zip over here. How about if I catch her first? She's closer.
I have a student that might be interested in forensic sciences. So wondering what UCs or universities in California might have the highest available technology or education. Maybe not in California.
Yeah, I think so. The most important thing, independent of forensics, is to get a good science foundation. So, and I would say any kind of science is interesting to them. So chemistry, biology, math, that's really key. The forensic science course of study is sort of layered on top of the normal sort of university study. And UC Davis has a forensics program. It's actually the only accredited forensics program in the state right now. But there are a number around the country and other programs in the state for forensic training, depending on how far the student wants to go. So a lot of great programs. We love to have interns a little more. We bring in summer, students in the summers, college students in the summer, and graduate students, and so we have a pretty active program of at least having part-time students come in during the summer as well.
Yeah, I would just make just one more comment about that. I think what you're seeing in forensics is really a turn away from a lot of the more traditional approaches. And so as Brad said, I think there's going to be a lot more science impacting forensics in the immediate future. And so I would just like to amplify what he said. I think the big impact in forensics, I think, is going to come from people who understand science and understand new ways to approach these kind of traditional forensics problems. So I think that's really where the big future is going to be.
For a person who would be interested in following up on either all or part of what this is, because they have an interest, what journals and or websites would be the most appropriate to access to, and besides the Lawrence Livermore Lab, of course, what would be the most 3 or 4 top websites or journals to read that are available?
Journals, and so this work has been published in a great journal that's online. It's an online journal, and it's available to the public. It's called PLOS1. It's the public, what is this?
Library of Science.
Public Library of Science. So this work has been published there, and there's a lot of great cutting-edge science in there. And the other organizations that have a real interest in this are actually NIST, the government agency NIST, has a great forensics set of programs and very good information on their website. And National Institutes of Justice also. So most of the work that's done in forensics is supported by the government through government research funding. And those are the main organizations that do that. And of course, there's the Journal of Forensic Science for the normal forensic science journal. Yes, sir.
I was interested in how you identify individuals with their DNA. I remember something about SNPs, and that's the way that they used to identify individuals. Did they not use that anymore?
So very early on, SNPs are single nucleotide polymorphisms, and we talked about those in the talk a little bit because it's those mutations of those SNPs in the DNA that cause the mutation that we look at in the protein. But you need a large number of them, and the same issues that we suffer from, you need more markers to get the same level of mutation. So that quickly transitioned to this STR approach because the potential power of discrimination, the random match probability you get, is so much higher with a fewer number of markers. So that's really the gold standard now.
So a follow-up, the people that do the genome, like 23andMe,
sure.
Is that the process they're using now?
They're looking at, well, so they're not doing human identification. They're doing, so SNPs are very important medical community, because those same mutations can be associated with disease or propensity for disease. So we think about things like the BRCA gene for breast cancer, for example. So 23andMe will use SNP panels to understand your genetic history and all the things that they promise you on the radio. Like, you can't remember all the things they do if you like to, if you're going to be able to run faster or whatever they say. So, or if you have potential for specific diseases and that sort of thing. So they do a lot of SNP, looking at SNP panels.
And just to add to that, so because we are seeing the reflection of SNPs in proteins, a lot of that information that you might find in a SNP panel is potentially available to us as well. And so, you know, the marker, your SNP markers in your hair reflected in amino acid changes could be indicative of your genetic background from kind of an ancestry point of view as well. And so that information will also be available.
Another question? Oh, right here, and then I'll be over there. Okay.
How does DNA indicate an area of origin? I've seen advertisements about you're coming from Scotland or you're from this part of Africa. How does DNA indicate an area of origin?
That's a great question.
Yes, so it has, well I think they're basically finding correlations between particular mutations, and there are these SNPs that this gentleman was referring to. And so if you look at the DNA, the particular mutations, and if populations are isolated, those mutations would be common to that particular population. And so you can find a SNP marker, you know, because this group had lived together for a very long time. It's common in that group, but say absent from another group. And so what 23andMe has done is taken larger and larger collections of these SNPs that are indicative of different parts of the world, and looking for which ones are present and which ones aren't, and particular combinations that give you, even more information about that. But it's those SNPs, that signal nucleotide polymorphisms. And again, what's so interesting about, I think what we're doing is those SNPs are responsible for these amino acid changes in our identity markers. So a lot of that correlated information is also carried in the proteins.
Has there been any talk of using protein analysis on ancient humans? We have like well-preserved examples of Neanderthals or woolly mammoths, things like that.
Yeah, I mean, so it's a great question. Deanne talked about the applications in archaeology, and so we've looked at hair samples that were, what,
200 years old.
250, 200 years old, that were from the UK, from England. There's a lot of information that you can get there that can help archaeologists, and so specifically in human populations, they want to understand migration patterns. Where did these ancient populations come from? And sort of tracing that back based on information that's available in the protein, because DNA often isn't available anymore in these remains. You see sort of like the Jurassic Park and stuff, that's not reality most of the time, right? But the protein is persistent, and hair in particular. And so being able to basically see an echo of the DNA of a person that's left behind in their protein when the DNA is gone has incredible value for things like that. So yeah, archaeology is going to be a huge benefactor of a lot of the work that's going on.
Yeah, for me, a particularly interesting example of this is kind of timing first European contact with South American native populations. And that, those European SNPs, as we talked about, that you might be characteristic of a European population, start showing up in the native populations. We learn something about those first contacts and the timing and so forth. So those are all very interesting. Under many conditions, hair can survive for very, very long times. We should be able to pick out those kind of markers in those archaeological samples and answer questions about migration and contact.
Any other questions? Oh.
Have there been any long-term comparative studies of an individual's DNA over time? Like, is my DNA the same as it was 24 years ago when I was born?
Yeah, that's a good question. In the context of hair, that's something we're very, very interested in, because there are the question is with age, is the way your proteins are expressed in your hair when you're young the same as when you're older? Certainly your hair has changed, right? It's gone from, just changed colors, changed texture, and so forth. The DNA should not have changed, but you really need to do the study that shows that. I think the STR regions for identification are stable in an individual, so the STR-based identification is. For our research, we're very interested in these protein markers with age of a person, also with age of the hair, right? And for example, if your hair is very long, the hair on the end has been exposed to the environment for a very long time, so there's your hair that's very close to the scalp. Also, you have hair on different parts of your body, and are those all reflecting the same information? And so that's part of our ongoing research right now, is to do that kind of work to understand age of hair, both from an individual point of view, age in terms of how long it's been in the environment, different location on the body, different hair treatments, right? Does dyeing your hair interfere with the type of analysis we're doing? And so those are all kinds of questions of ongoing research that we're working on right now.
Anyone else? Oh, got one back here. Two.
Would you be able to determine chronological age from the proteins in the hair?
We haven't thought of a way yet, but that's a very interesting question, because certainly, like I said, the outward appearance of your hair has changed. The question is, has anything in the protein signature changed? You would guess that the type of markers that we're looking at, which are based on your DNA, probably have not changed with age. But I said probably, right? There may be data.
There's also a question of you may need to know, it's very individualized, right? So how is a single person, this person's hair changed over time? So you need to understand what the starting point was and how has that changed to have a benchmark. But no, it's a great question. I don't think it's something that we really wrap our arms around here.
Okay, so you talked about the short tandem repeats and how they're a more objective method of identifying an individual. So to what extent is that a reliable and accurate way of actually determining a person? Because is it always different from person to person, and is it always varied?
It's incredibly powerful, and so and we know that because we have statistics. We understand for very large populations what the frequency of those STR regions and how they change between people. And so by knowing that math, we can do what the process that Ian showed in Orbit with the developing the product world, and we get incredibly high, what they call random match probability. So the probability that we will randomly match some other person with that same sequence is super low. It's much, much lower than the entire population of the planet. It's actually much lower than every human that's ever been on the planet. Okay, so the precision that you have there is incredible.
Any other questions? I think we've exhausted our audience.
Thank you guys so much. Wonderful question.
We also have a